Flow cytometry is the measurement of cell properties using light as cells pass in a fluid medium. The system has several major components. At its core is the laser, or light amplification by stimulated emission of radiation. Most commonly, an argon gas laser is used, which emits light at 488 nanometers. Beam blockers shield the detectors, while those detectors capture photons from disturbed light or from fluorescence signals produced by stains. The cell stream itself is carried in a flow cell, which aligns cells single file. The principle is straightforward. When cells pass through the laser beam, they disrupt the light and create signals that reflect their size and complexity. The laser also excites fluorochromes, fluorescent dyes that are attached to antibodies or directly to the cells, producing characteristic light signals. Two kinds of scatter are especially important. Forward scatter depends on diffraction and refraction, and it primarily reflects cell size. Side scatter, detected at 90 degrees to the beam, depends on reflection, and it is sensitive to cell granularity and internal complexity. Before white cells are analyzed, red cells are usually lysed away. Using forward and side scatter signals, the instrument can separate populations, such as lymphocytes, monocytes, and granulocytes. These populations can then be electronically gated for more detailed study. Fluorochromes are central to immunophenotyping. Direct fluorochromes include FITC, which fluoresces green between 500 and 530 nanometers, and PE, or phycoerythrin, which fluoresces yellow between 550 and 590 nanometers. More advanced tandem dyes include PE Texas Red, PE Cyanine 5, and PE Cyanine 7, which extend the range into orange, red, and infrared. These dyes are conjugated to monoclonal antibodies directed against specific cell surface markers. In HIV monitoring, for example, antibodies are used against CD45, present on all leukocytes, CD3, on all T lymphocytes, CD4, on T helper lymphocytes, CD8, on T suppressor lymphocytes, CD19 on B lymphocytes, and CD16 or CD56 on natural killer cells. Ensuring antibody saturation and verifying with quality control are essential steps. Analysis can be done with single-color staining, but this has largely been replaced by multicolor analysis. In two-color analysis, for example, FITC and PE may be combined in the same tube to distinguish subsets like CD4 and CD8 T cells. CD45 and CD14 can be used together to gate leukocytes and exclude monocytes or debris. 3- and 4-color panels build on this approach, allowing simultaneous detection of multiple populations, such as CD3, CD4, and CD8 in the same panel. Flow cytometers also provide quadrant statistics, calculating the percentage of cells in each region of a plot. Absolute counts can be derived by a dual platform method, using results from both the flow cytometer and a hematology analyzer, or by a single platform method using calibration beads within the cytometer. In HIV monitoring, CD4 counts are critical. A normal CD4 count ranges from about 450 to 1500 per microliter. When counts fall below 500, prophylaxis for opportunistic infections may be considered. AIDS is clinically defined as HIV infection with CD4 counts below 200 per microliter. Beyond HIV, flow cytometry is widely applied in immunophenotyping leukemias and lymphomas, detecting fetal cells in maternal circulation, and supporting stem cell transplantation through detection of CD34-positive stem cells. In summary, flow cytometry is a powerful technology that combines lasers, scatter signals, fluorochromes, and monoclonal antibodies to provide highly detailed cellular analysis, essential for both diagnostics and monitoring.